Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR Net JRF Life Science question paper. This is shift 2 questions that I am discussing, and today I will be discussing the cell biology questions. This was conducted in the year March 2025. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of CSIR being discussed, you can go to my channel Omni Bioentrance. In the channel playlist, select for CSIR Net exams and there you will find. So coming to the first question, a putative mitochondrial signal peptide was attached to the N-terminus of the DHFR protein and expressed in mammalian cells. So DHFR, I think it is dihydrofolate reductase protein, and that was an N-terminal signal was attached to this DHFR, and it was targeted for expression in mitochondria. Okay. So the N-terminal sequence, if it is targeted to mitochondria, it will be amphipathic alpha helical in nature. Okay, this N-terminal sequence. That's just for your information, I told, that's all. Then mitochondria were isolated and a fraction was osmotically shocked briefly. Okay, so mitochondria, they are isolated and a fraction of it was subjected to osmotic shock treatment. Now this osmotically treated mitochondria as well as untreated mitochondria were treated with protease. Okay, first they were given osmotic shock and then later it was subjected to protease treatment. It was observed that the DHFR was intact in both the cases. So DHFR was not affected, that means it has entered mitochondria. Now where it is localized, you have to find out from the options. Okay, which of the following statement best describe the function of signal peptide? Now where is this signal peptide targeted? Whether it is to the outer membrane, to the intermembrane space, to the inner membrane or to the matrix, you have to tell. So you know that mitochondria, it is having an outer mitochondrial membrane and there is an intermembrane space here, and then you have got the inner mitochondrial membrane which is folded into cristae. There are so many folds for that, and then you have the matrix. Okay, so what will happen when mitochondria are subjected to osmotic treatment? So this treatment, it will disrupt the outer membrane. So this membrane, that is the outer layer, it will be disrupted and it will make the intermembrane space, the next layer is the intermembrane space, accessible to proteases. But the inner mitochondrial membrane, it will be generally resistant to osmotic shock because it is folded into cristae. It is highly folded, so it will be resistant to osmotic shock. And what does this protease do? It will digest the protein that are exposed to the cytosol or present in the intermembrane space. Okay. Now, what is that you observe? You observe that the DHFR is intact in both osmotically shocked and untreated mitochondria when treated with protease. Okay, so it is intact whether you treat with osmotic shock or you don't treat the mitochondria with osmotic shock, and then you treat with protease for both the mitochondria, your DHFR is intact in both the cases. So what do you understand from this? So first statement, it says that it targets DHFR to the mitochondrial outer membrane facing the cytosol. That is, this is the mitochondrial membrane. Here you will have your cytosol. Now DHFR is facing the cytosol. That is what they have mentioned. And the signal peptide, it is targeting the DHFR to the mitochondrial outer membrane. That is what they are telling. Now if DHFR is targeted to the mitochondrial outer membrane and is facing, and its domain, that is the DHFR domain, is facing the cytosol, then In that case, it should have been digested with the protease. Okay, that is in both untreated, that is if the untreated mitochondria, that is which was not subjected to osmotic shock, as well as the mitochondria which was subjected to osmotic shock, would have become susceptible to DHFR with protease treatment. Would have become susceptible to protease treatment. The DHFR present would have become susceptible to protease treatment. But what they have observed, DHFR is intact. So first one cannot be the answer. Then it targets DHFR to the mitochondria inner membrane facing the intermembrane space. Here also the same story. Now if the signal peptide has targeted the DHFR to the inner membrane, that is over here, and its domain is facing the intermembrane space, then osmotic shock treatment. Osmotic shock means outer membrane got disrupted, the DHFR got exposed to protease, leading to its digestion. It would have taken place in the case of treated mitochondria. But here the DHFR is intact in both the cases, so it is not to the inner membrane that DHFR is localized. Then that also wrong. Then it target DHFR to the mitochondrial outer membrane facing the intermembrane space. Now if DHFR is embedded in the outer membrane and the domain is facing the intermembrane space, then osmotic shock treatment, that is disruption of the outer membrane, would have exposed it to protease, resulting in digestion. But the observation is DHFR was intact, so that is also not the case. Then it targets DHFR to the mitochondrial matrix. Yes, that is correct. Now because DHFR is protected from protease even after osmotic shock, that is after disruption of the outer membrane and the exposure of the proteins on the intermembrane space, and then you treat with the protease, DHFR was found to be intact. So this suggests that it should have been in a compartment which is inaccessible to protease. So naturally what is left out? It is your matrix. So the answer is option 4. It targets DHFR to the mitochondrial matrix. Then next one. The lipid composition of two monolayers of the plasma membrane is quite different. Okay. The lipid asymmetry is functionally relevant, especially in converting extracellular signals into intracellular ones. Given below are a few membrane lipids. Now you have to say where these are localized. So choose the option that correctly defines all the lipids involved in signaling and are restricted to the cytosolic phase of the plasma membrane. That is important. So where you have to see for lipids which are localized on the cytosolic phase of the plasma membrane. So phosphatidylserine, yes, it is on the cytosolic. See here, phosphatidylserine, a cytosolic phase. Phosphatidylinositol 4-phosphate, phosphatidylinositol, it is again on the cytosolic side. Phosphatidylcholine, extracellular, not on the cytosolic side. Sphingomyelin, again extracellular, not on the cytosolic side. So your answer is A and B. That makes option 2 as the answer. Now next one. Cholera toxin activates the chloride channel in the epithelial cells of the intestine, leading to loss of chloride ions and consequent loss of water, leading to dehydration. Okay. Successful oral rehydration therapy involves supplementing water with glucose and salt and not just salt. Why is it so? Now we will see the mechanism of action of cholera toxin. Only then you will understand what is happening in oral rehydration therapy. So cholera toxin produced by Vibrio cholerae is having two subunits, A and B subunit. The B subunit will bind to the GM1 ganglioside on the epithelial cell of the intestine and it will be internalized. First it will enter to the Golgi apparatus and then to the endoplasmic reticulum. In the endoplasmic reticulum, the A subunit will be dissociated into A1 and A2. The A1 peptide, it will enter into the cytoplasm from the endoplasmic reticulum lumen and it will interact with the G protein. G protein, you know, involved in cellular signaling. It will activate adenyl cyclase and result in the production of CAMP. So same thing is happening here. The A1 peptide, it acts as a catalyst to ADP ribosilate the G protein, causing it to be constitutively active, and it will activate the adenyl cyclase and it will result in the increase in the production of CAMP, and CAMP is a secondary messenger, and the CAMP levels will activate the cystic fibrosis transmembrane conductance regulator, and it is a chloride channel, and this will result in the secretion of the chloride ions from the intestinal epithelial cell into the lumen of the intestine, and along with the chloride ion, sodium and water also will go out into the lumen, and this will result in loss of fluid or dehydration in the patient, and it is the cause for the diarrhea, watery diarrhea in cholera affected patient, and they have rice water stool like appearance which is associated with cholera infection. Now what is it that you suggest usually for a cholera infected patient is you give them oral rehydration solution or ORS. Now ORS, it is having a precise balance of glucose, sodium, potassium as well as other small electrolytes along with water. So what is the principle behind this ORS? So ORS, it utilizes the glucose and sodium sym-porter mechanism or the co-transport mechanism of the small intestine to facilitate water absorption. Now how is it done? So when ORS is taken inside, the glucose in the ORS solution is absorbed in the intestinal cells. Along with that, sodium will also enter. Okay, because of that, now sodium and glucose has entered, water cannot stay happily outside, it will also enter. So thereby it will start rehydrating the patient. Now without glucose, sodium will not be absorbed by the intestinal cells. So that is why this ORS or your oral rehydration therapy, you always include both sodium and glucose. Okay. Now come to the options. So to replenish the energy lost by dehydration and replace chloride. No, it doesn't replace any chloride or anything like that. So that is not correct. To create an osmotic gradient with Na plus and glucose to allow water to move from intestinal lumen to blood via epithelial cells. Yes, that is correct. So that is what it is doing. It is creating an osmotic gradient and Na plus and glucose will enter into the intestine, allowing water to enter and rehydrate the patient. Now because the epithelial antiporter pump, it is not antiporter, it is sym-porter or co-transporter. So that makes 3 also wrong. Because sodium chloride will release Cl minus. Sodium chloride is not splitting and giving out Cl minus ions and replacing the chloride ion. The principle is different. Sodium and glucose will enter into epithelial cells. Along with that, water also will enter, rehydrating the patient. So that is not correct. Now next one. After synchronizing mammalian cells in culture with a double thymidine block, cell cycle duration of 24 hours, cells are released into fresh medium. What is this double thymidine block? Now, thymidine, you know, it is essential for DNA replication. What will happen when you expose the cell to high concentration of thymidine? It will start inhibiting the enzyme thymidine kinase. And this thymidine kinase, it is essential for DNA synthesis. So what will happen as a result of this? Thymidine will start accumulating in the cell. And when it keeps on accumulating, it will halt, or I can say it will stop the DNA replication. So when you apply thymidine block, the arrest for the cell cycle is taking place at the G1S phase, S boundary of the cell cycle. Okay, at the G1S boundary of the cell cycle, the arrest is taking place. Okay. So when they have told it is double thymidine block. So first you apply one thymidine block, and it will stop the cells from going to G2 at the early S phase as well as in the G1 phase. So there are some cells which are in the mid S phase and late S phase also. They also have to be brought and blocked. Only then you will get a homogeneous population of cells which are arrested at G1S phase. So for that you apply a second thymidine block. So this second thymidine block, it will make sure that the cells in the mid and the late S phase are also blocked or arrested, and you will get a homogeneous population of cells which are arrested at the G1S boundary. Okay, that is why you apply this double thymidine block. Okay, now what you do after that, after some time period, you will remove the block and thereby release the cells into a fresh medium. So here all the cells are synchronously dividing when you are putting them in the fresh medium. All from G1S, they will enter into the next cell cycle stage G2, then M. So all are synchronously dividing. Okay. So what do you learn by doing all these things? You can collect data at or the researchers can collect data at what events are taking place at specific phases of the cell cycle, like what is happening in prophase, what is happening in metaphase, what is happening in anaphase, what will happen when you apply some inhibitor at anaphase, what will happen that time, what will happen if you apply an inhibitor at the metaphase stage. Okay, all these things you can learn when you do a double thymidine block, and then you remove the block so that you get a synchronously dividing cells, and you can learn all these kind of things after removal of the block, or the various stages of the cell cycle also can be studied. Now what are the different stages of the cell cycle? So prophase, where you find that the chromosomes have become visible, and spindle formation have just started to take place. There is a breaking down of the nuclear envelope. Then comes the pro-metaphase, where the nuclear envelope is completely broken down. The spindle formation is very prominent, and the spindles have started to attach to the kinetochores of the chromosome, and they have started to align on the metaphase plate. What is happening in metaphase? The chromosomes are fully condensed, the spindle fibers are attached to, and the chromosomes have aligned in the metaphase plate. And what do you see in the anaphase? sister chromatids got separated. They are moving to the opposite poles by pulling or shortening of the microtubule. Then comes your telophase, where the nuclear membrane have started to reform and the chromosomes have become to uncoil. Then this is followed by the cytokinesis, where there is a cleavage furrow formation and the division of the two daughter and the formation of two daughter cells. This is what is taking place in telophase and cytokinesis. Now what are the various cyclins that are getting activated at the various stages of the cell cycle? So if you see at G1 it is cyclin D, at S phase it is E mainly, and at G2 and M phase it is cyclin B that is more prominent, and in G2 phase cyclin A is also there. So yes. So G2 M phase it is cyclin B that is more active. Okay. Now come to the question. So they have released after removal of the thymidine block. So you get a synchronously dividing cell. So after 6 hours, cells are split into 4 sets and each is treated with nothing or proteasome inhibitor or myosin 2 inhibitor or nocodazole. Okay, now after 6 hours, so this is a cycle of 24-hour duration. So after 6 hours, it will be mostly in the G2M transition stage of mitosis. That is, your cyclin B is getting activated, it's being produced. So it is mostly in the G2M phase. That is, they have started to enter mitosis there. Now when you treat the cells with nothing, you get a synchronously dividing cell. Nothing else, no treatment. What do you see? Synchronously dividing cell. So that makes 1, 2, okay, 4 also taken, we have to take into consideration, but 3 they have told it is binucleate cell. That's not correct. Then proteasome inhibitor. Now where is this proteasome coming in the cell cycle? At what stage of mitosis? So G2M transition, I told you it is your cyclin B that is rising, or it's increasing in concentration. Now proteasome, they are involved in the degradation of these cyclins. So cyclin B has to be degraded for the cell to exit the mitosis, as well as for the cell to enter from the metaphase to the anaphase. Now cyclin B degradation, it is mediated by the anaphase promoting complex, and this will tag the cyclin B with the ubiquitin, and it is targeted for proteasomal degradation. So that is where your proteasome is coming. So when proteasome inhibitors are applied, degradation of cyclin B will be blocked. Okay. So as a result of that, cyclin B levels will keep increasing, and it will prevent the exit from metaphase and entry into anaphase. Okay, now cyclin B has to be degraded for metaphase to anaphase transition also. Not only for exiting from mitosis, but also for entry from metaphase to anaphase. So the cyclin B proteasome is inhibited, means cyclin B levels are increasing and the cell will not be able to progress from metaphase to anaphase. It will be blocked at the metaphase stage. So that makes option B, option 1 not correct, because they have told it is binucleate. Then 2 also, it is not at prometaphase, it is at metaphase that it is arrested. So that means 4 is your answer. Now next one, myosin 2 inhibitor. Now in mammalian cell division, myosin 2, it is involved in cytokinesis. That is in the formation of a contractile ring that drives the cleavage. And the contractile ring, it is composed of the actin and the myosin 2 protein. Now, so myosin 2 is inhibited, means cytokinesis will be prevented and you will get binucleate cell, that is those cells which have completed mitosis but fail to divide because cytokinesis is not taking place. So you will get binucleate cell. Then nocodazole, it is a microtubule depolymerizer. And that means tubulin dimers are removed from the ends of the microtubule, causing it to shorten. That is what nocodazole is doing. So nocodazole, it will arrest the cell in the pro-metaphase, because in that stage only the microtubules are more prominent and they are polymerizing, okay, in normal cell division. So at that stage the cells will be arrested if you treat them with nocodazole. So answer is option 4. Then a region of a mouse chromosome was subjected to micrococcal nuclease hypersensitivity analysis over stages of development. Now micrococcal nuclease, it is an enzyme that digests DNA. And it will cut the DNA at the linker region between nucleosomes. Okay. So in early stages the region had regularly spaced nucleosome, whereas in later stages nucleosomes were irregularly spaced with several nucleosome free region detected. Now what is your observation? Now early stages the nucleosomes are regularly spaced. So after a little while, after treatment with the micrococcal nuclease, you can see that the nucleosomes are irregularly spaced and there are some nucleosome free regions, that is the DNA is not bound to the nucleosome, it is free. So if it is not bound to the nucleosome and if it is free, means it is accessible to your transcription machinery and there will be expression of genes. Okay. Now what is the best possible inference for this? Okay. Now you all know that chromatin, there is euchromatin and heterochromatin. Euchromatin is loosely packed, it is transcriptionally active, and the cells will produce protein from this region, because the loose structure of the euchromatin make it easily accessible to various transcription factors and other transcription machinery. Whereas heterochromatin, you know, it is tightly packed and it is inactive and it is inaccessible to transcription factors and transcription machinery. Now heterochromatin, it is of two types, that is constitutive and facultative. Constitutive means whatever you do, it will remain in its condensed form and it will always be inactive. Whereas facultative heterochromatin means, yes, it can at times get converted to euchromatin also, but it is facultative and it can remain in heterochromatin stage also. So if you apply certain conditions to a facultative heterochromatin, it can get converted to euchromatin and make a transcription machinery accessible to it and result in gene expression. Okay. Now in this question, you can find that at early stages it is regularly spaced in nucleosomes. So it is like heterochromatin, regularly spaced, packed, not accessible to transcription machinery. But after some time, after treatment with the micrococcal nuclease, you find that is what is meant by in later stages, the nucleosomes irregularly spaced. And there are free regions also, that is without nucleosomes. The DNA has is getting access to the transcription machinery there. So it must have been previously it must have been a facultative heterochromatin, and because of micrococcal nuclease treatment it got converted to euchromatin. So the chromatin region is a facultative heterochromatin, that is correct. The region is highly expressed in early stages. No, in early stages they have told it is regularly spaced in nucleosome, it is packed. It is heterochromatin at that time, not euchromatin way. So that is not correct. Then nucleosomes are not made efficiently in the late developmental stages. No, it is not like that. Because micrococcal nucleus, they digest DNA by cutting in the linker region between nucleosomes. So it doesn't say that nucleosomes are not efficiently made. So that is not correct. Then nucleosomal arrangements cannot be used to infer potential expression status. That is also not correct. Then next one, given below are a few statements about vesicular transport. Okay, the vesicular transport I discussed in part B question, where we had discussed about the V snare, T snare mechanism. Now this here it is about the receptor mediated endocytosis. Okay, now we will see what is this receptor mediated endocytosis, and I will be discussing the LDL one. Okay. Now you know that in mammalian cells the uptake of cholesterol is the key model for understanding receptor mediated endocytosis, and cholesterol is transported through the bloodstream in the form of LDL, not as cholesterol. So how does the cell utilize cholesterol? So LDL has to be taken up by the cholesterol. Now for that there is an LDL receptor. You can see here. This is the LDL receptor. This is the extracellular domain. This is the end terminal of the LDL receptor to which the LDL will bind. This is the transmembrane region of the LDL receptor, and this is the cytoplasmic domain. Now to this this is the receptor in clathrin coated pits. Now the cytoplasmic domain, it will bind to the adapter protein. See here, AP2, that is the adapter protein. It will bind, and this adapter protein will bind to the clathrin. These are the clathrin. Okay, so it will bind there, and it is facing the cytosolic side. So now when the LDL is bound to its receptor, it is internalized in clathrin coated vesicle, and later the clathrin coat is shed off, or it loses the clathrin coat, and it fuses with the early endosomes, which has an acidic pH of around 6 to 6.2. These are the early endosome, and because of this acidic pH, the LDL, it dissociates from its receptor. Okay, and the receptor is recycled back into the plasma membrane, and it is ready to accept a new round of the ligand. Now what happened to this LDL? Now LDL, it is transported from the early endosome to the late endosome. Okay, now that picture I couldn't get it in that way. Okay, but I will tell you, there are transport vesicles that are carrying the lysosomal hydrolases from the Golgi apparatus. Transport vesicles carrying lysosomal hydrolases from the Golgi apparatus. These will fuse with this late endosome, and these late endosome upon fusion will mature into lysosomes. Okay, and in the lysosomes, the LDL will be degraded and cholesterol will be released. That is here. Okay, cholesterol is released, which can be taken up. Now, and I told you LDL receptor is recycled back. So that is what is happening in LDL receptor mediated endocytosis. Now come to the question. So clathrin mediated endocytosis requires the requirement of adapters to the cytosolic phase of the plasma membrane. I already told you that on the cytoplasmic tail of the LDL receptor, adapter protein will bind, then the clathrins will bind. So A is correct. The low pH environment of the early endosomes, yes, lead to the dissociation of the cargo from its receptor. Cargo is your LDL. So LDL will be dissociated from its receptor, yes, allowing the recycling of receptor to the plasma membrane. That is also correct. Then the late endosomes which mature into lysosomes are directly involved in the recycling of synaptic vesicle proteins in neurons. Okay, now before answering that, I will tell you what is happening to the synaptic vesicle proteins in neurons. Now this is the synaptic vesicle in a neuron. This is the synaptic vesicle. It has acetylcholine transmitter molecules. Now this is your presynaptic membrane and this is your postsynaptic one. So once an action potential is generated in the presynaptic terminal, it will result in the fusion of the synaptic vesicle to the plasma membrane of the presynaptic membrane and will release of the neurotransmitter into the synaptic cleft. Okay. Now the whole of the neurotransmitter within a synaptic vesicle will be released and the empty synaptic vesicle has to be recovered back so that it can be refilled again with the fresh transmitter molecules or acetylcholine. So for that, these empty synaptic vesicles, they are recovered from the plasma membrane in clathrin coated vesicles. See here, clathrin coating. Okay, clathrin coated vesicle, and these are the clathrin coated vesicle and they fuse with the early endosome. In the question they have given it is late endosome, which is not correct. It is early endosome, and these synaptic vesicles, they are regenerated by budding off from the early endosome, and then they are accumulate, or they will accumulate new neurotransmitter molecule and they can again enter into the cycle, so for the next cycle. Okay, now see the question. So the late endosomes, so A and B correct, which mature into lysosomes are directly involved in recycling synaptic vesicle proteins in neurons. If you don't know this concept also regarding synaptic vesicle protein recycling in neurons, if you use or if you think properly, you will find that late endosomes mature into lysosomes. Lysosomes are what? They will degrade of whatever enters into them. So the synaptic vesicle proteins, they will also be degraded, which is not happening in your neurons. They are recycled again. So that makes the question C as wrong. That means 3 gone from the options. Okay, now we have to see the D one. The multivesicular body pathway involves the formation of intraluminal vesicle which sort cargo for degradation in lysosome. Yes, that is correct. So the multivesicular body, it is also playing a role in receptor mediated endocytosis, and it is mainly for the degradation of the internalized receptors, and it also involves the formation of intraluminal vesicles within the endosomes, and which mature into multivesicular body and which fuse with the lysosomes for degradation. Okay, so A, B, D are correct. That means 4 is the right answer. 1 and 2 are incomplete. So A, B, D are correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, then if you want still previous year question papers of CSIR net been discussed, you can go to my channel Lumini Bio Entrance, and in there you will find the playlist. In the playlist you can find CSIR net exams, and in there you will find. Thank you.